Good morning, good afternoon, everyone, and a warm welcome to this breakout session regarding data and scientific insights for treatments of COVID-19. I am joined by an outstanding group of speakers and panelists. If we go to the next slide. And just to say, I hope that you did enjoy the plenary session, barring some of the logistical issues. And we are going to try and make our breakout just as informative and just as exciting as the content that you caught in the plenary session. So you'll see we have speakers from the ICODA team. Joining me, Nevine, is Jonas Haggstrom, Jaap Mandema. And then a little later on, we are going to invite our collaborators, Phil Ambery from AstraZeneca, Ben Kramer from Genentech, and Peter Mesenbrink from Novartis. Let's go to the next slide. As we go through at any point in time, questions and comments are very welcome. You'll see me trying to catch as many of those as I can. So please feel free to jot them in. And if not now, if questions come to you tomorrow, believe me, this is an open-ended invitation to get in touch with us. We'll cover first off more insights into how the Data Research Lab is being constructed. We'll talk about the tools, the data assets that are available now and soon to be available. I think you'll find that interesting. And Jaap will take you through a very specific example of what you could do now, which hopefully will spur the imagination as to other research questions you may have. And we'll go straight into a panel discussion with Q&A thereafter. Let's go to the next slide. So you saw this in the plenary, and just to remind everyone, we have a very ambitious goal, and that is to use data as the fuel to unlock scientific insights, which in turn will help us address this pandemic worldwide. Now, if we do our work well, not only will this infrastructure and approach be relevant for this pandemic, but for future pandemics, as well as any other health research questions. So I engage you to think about that as well. Next slide. So I'm going to take you through 3 slides that really talk about the infrastructure that we have from a technology standpoint. And this first slide puts it in the lens of, I'm a data researcher, and what is it that the ICODA workbench does for me? Well, first, it will provide you the ability to search relevant data sources from one spot, no matter where these data sources are around the world. So that's a pretty big advantage right there. Second, you get to request the data access through a single mechanism, as opposed to having to learn individual little peculiarities of every data organization. Third, as you heard from Neil in the previous panel, we actually have provided a range of tools and a secure environment within which to use these tools. And then in the case where data cannot travel, as in, for example, the Sentinel kind of system, your analysis can travel to the data and bring you back the results in the workbench. And then the ability to work in collaboration with others, either sharing your code, borrowing their code, talking about results, features of the data, etc., is something that is very much enabled within the workbench. So that's if you're a data researcher. Let's go to the next slide and think about it in terms of those of us who are concerned about security, governance, and maybe we're a data organization. We're wondering just how reasonably set up this infrastructure is. So first, just to know that it's scalable, it's cloud-based, it's secured, it allows multiple project teams to work together in their workspaces, their private workspaces, at the same time. And we've already thought ahead to the data models that allow for this work to happen in a free flow but informed sort of way. Now, if we're thinking of collaboration, then we're thinking open science. You probably figured that out from the prior conversations. So that means state-of-the-art tooling when it comes to onboarding of the data and making it verified. And then the other piece is let's use tools that are out there. And Neil again mentioned a couple of tools from our collaborator Cytel. They're actually strategic partners, I should say. That are now available for anyone to use within the workbench. And this is just the first of many, many, many options. We already have another partner, Certara. We'll see that tool in action for us today. And so you could see how a small investment from one partner then grows into something much bigger for all collaborators. And then we've adopted GPRD as our security and governance system. It is the most complete of the models out there. So I think that's reassuring, complete traceability and audit of work that's done within the workbench as well. So let's go on to the next slide. The last piece that I wanted to flag for you is this question of what are the ways within which data contributors can provide their data so that they control the level of access and the means by which access is granted. So first you have the model that we have adopted and that our pharma partners have adopted for randomized controlled trial data for treatments of COVID-19, and that is to have the data directly within the workbench. It's accessible directly within the workbench. That's a very substantial commitment that they've made to us, and we're very proud that we've garnered their confidence in doing so. The second is that the data stays in its originating environment, but the researcher can request it from them, and at the time access is granted, the data is ported to the workbench to sit alongside all that other data that we've described. So that's what we call level 1. And then level 2 is when the data stays in the satellite organization and the analysis code travels there and brings results back. And the trick here is that we allow all of these to be available at the same time so that the data researcher can do their work in the one workbench with the data being provided in this plurality of ways. So that is a bit more detail on the actual workbench, the infrastructure, the tooling, the data access, the data models. Let's go on to how you make a thriving community happen. Next slide. You saw this earlier. I was going to give it perhaps a slightly different commentary for all of us. So first is, it's got to be global. There is great scientific work going on all over the world, and we want to benefit from that holistically, number one. Number two, not a single one of us, no matter how great our ideas are, has the best ideas. So we must collaborate, and we must be humble in knowing that other ideas may be just as good or better than ours, and together we build something that is truly something we can be proud of. Third, if we want to facilitate open research, we really have to do some basic work. So the interoperability is so easy to say and quite hard to do, and we want to do it at scale. So we need our technical experts to really come out of their silos and bring us the best, the smartest, the most clever, the most modern approaches. And we've got the start of that already. We'll be hopefully publishing on that in the first half of next year. Trust, you don't get anywhere without trust. And then here's the thing that I like the most. As you can tell, we've done quite a bit of work. We're ready, open for business. And then we push through the research projects, the driver projects that Aziz mentioned. And that's what we're going to spend the rest of our time today talking with you about. Next slide. So what did we do? We took a survey back in July. We saw where researchers, policymakers, other stakeholders were most interested. And we set the very first research project, we call it Driver Project 1, to look at these features of safety and efficacy of treatments of COVID-19. So having done that setup, I am going to hand over to Jonas Haggstrom to tell you more about Driver Project 1. Jonas, over to you.
Thank you so much, Nevine. To continue what Nevine said about Driver Project 1, what data do we have in the workbench and what trials are we going after? To start with, we are looking at randomized controlled clinical trials. As you heard Martin Lander said, I mean, this gives the highest level of evidence are well performed as well. In addition, we're also looking at platform trials, especially graduated arms. And we see no issues with integrity of the trial by going after the platform trial. So now we are looking at the randomized controlled clinical trials. What data are we going to get from those trials? Well, we've started with summary level data. There's less concerns with privacy concerns with summary level data. It is ready to use and can start answering in a rapid way many of the research questions we're interested in. So now we have randomized clinical trials. We have agreement to get those. We have summary level data. How do we make the most out of using the summary level data? To do that, we started out with harmonizing and standardizing outcomes across the trials. By doing that, we were able to develop a data dictionary for COVID-19 summary level data, quite a novel data dictionary, I must say. I will show you a snapshot what is in there. We have also agreed with a data contributor to provide us data, summary level data, that goes way beyond what is available in the public domain. That will help us doing additional subgroup analysis. It will kind of help us standardize the times we are looking at it and so on. More or less looking at the sciences many different ways and many different ways we can do beyond in the public domain. So to summarize, we're going to get randomized clinical trials, enrich summary level data, and all of this will help us assess key questions, scientific questions of interest in a structured way. Next slide, please. So what is the current status? Currently, we have agreement from a dozen pharma companies. I must say it's an unprecedented agreement since they've committed us to give us enriched summary level data from the RCTs. We expect to have about 5 to 8 trials by the end of 2020. I mentioned platform trials. We have approached a few of them and we're making headway. Currently available in the workbench are Codex. Codex is a very good supplement to the summary level data, which collects public available data from public available resources. I must say Codex is an excellent tool to further investigate and framing scientific questions and for hypothesis generating. You will hear much more about that from Jaap in a few minutes. So all in all, we have randomized clinical trials, we have summary level data, we are making most use of the summary level data by harmonizing, standardize it, and we have, in addition to it, tools that will help speed up the guidance of almost real-time analysis. Next slide, please. So I mentioned the effort of harmonizing and standardizing the data we're going to get from the randomized controlled trials. This is the outcome of it. It's what we call the data dictionary. The general structure of the clinical variable is outlined on this slide. It's much richer. You can see in the actual data dictionary, but you get a flavor of the number of subgroup analysis you will be able to do, the number of efficacy points that are standardized and harmonized across the trials. And we have also tried to, for efficacy, safety, and exposure endpoints, define endpoints for baseline, day 3, 7, 14, and 28. All that will be helping us comparing like to like or apples to apples as much as possible. To further facilitate real-time analysis and so on. Next slide, please. We have visualization tools within the workbench. This is a snapshot or a screenshot of the visualization app. It works not only for the summary level data, but also for individual patient data. Everything is open code, and you can get access both within the workbench and outside the workbench. Next slide. To further facilitate interrogation of the data to answer scientific questions, we also have a meta-analysis app built in there that are developed specifically for the data structure given the harmonization standardization we have done. I think I'll stop there and hand it over to Jaap for an interesting journey through Codex.
Thank you, Jonas. Next slide, please. Codex is a database that contains curated and augmented publicly available information from studies that evaluate pharmacological treatments and risk factors for COVID-19 disease progression as well as treatment. And that information comes from a variety of sources, be it publications, pre-publications, press releases, the trial registries, wherever it can be found in a public domain. And it's rich with regards to the information it contains, meaning it has all the information on the population characteristics, concomitant treatments, any statistical analysis that was being completed on the data, as well as the results spanning efficacy, safety, biomarkers, and vital signs. Currently, there's about 60 randomized controlled trials in the database containing information representing 40,000 patients and 136 observational studies with information of about 150,000 patients represented through those studies. Apart from the data, there are tools to quickly zoom into segments of the data, to summarize it, to make graphs of it, to run some pairwise comparisons, and do exploratory network meta-analysis, all in an automated fashion on any of the endpoints that are represented in the database. Really, the idea is here to provide researchers with a quick snapshot of all the available information that's already out there from which they can form full research proposals going forward. Next slide, please. When you open up the app, this is the landing place. It represents its richness of about the 700 endpoints that are represented in the database. Death is the most commonly reported endpoint, and of about the 60 treatments that are represented in the current studies. Tocilizumab is the most studied, available in 37 studies, followed by hydroxychloroquine and others. You can tap through a variety of actions you can take with the data, and I'll show some of that later. And on the left-hand side, you can zoom in. You can pick particular aspects of the data that you might like to zoom into. For example, I may be interested in learning a little bit more about what studies on the next slide are available about treatments with corticosteroids. Next slide, please. So I select on the left-hand side, the corticosteroids is my treatment class, and I get a quick overview of the studies that are there with the most important characteristics of those studies. And I can look at each of the studies individually or look at them in a general overview and explore that a little bit further. And one of the things you may want to explore is let's, for that endpoint that's mostly reported, let's make a quick summary on the next slide of the available information across these studies. And I've picked death as the endpoint and representing the odds ratio, but obviously we could look at the risk ratio or the risk difference or the hazard ratio as well, if that is available. And here we've stratified the results or the trials by the randomized controlled trials on the bottom and the observational studies on the top. And if we just zoom into the bottom 5 studies that are the randomized controlled studies on steroids, we can see a general trend of about 0.74 odds ratio on aggregate across these particular treatments for death. Of course, I may want to zoom in a little bit more in the next slide. Maybe I'm interested in how this treatment effect is dependent upon the disease severity. So here I've stratified it by the disease severity group with the critical patients on the top and the mild moderate patients on the bottom to maybe evaluate if there's any trend of the effect with respect to the disease severity. And here you can already see where we run into the limitation of the publicly available information, namely that the stratification may not be available for each of these studies, or the definition that's being used to define the disease severity group is different across these studies, even though we try to harmonize that as much as possible. And that's where the enriched data sets come in that Jonas talked about earlier, to enhance the ability to detect these important interactions between the treatment modality and the course of the disease. On the next slide, I've shown a little more. Of course, you can add other treatments. I just restricted myself to the corticosteroids and looked at a different endpoint. Here it's critical disease, now represented by on the bottom, meaning patients that progress to being mechanically or invasively ventilated or dying. And for tocilizumab and the steroids, we both see a trend of showing some activity for this particular endpoint. And again, run into some of the limitations of the publicly available data. Here, the baseline status of the patients, meaning what level of critical disease they already have when they start the study, is quite different. That generates different interpretations of the results. Lastly, we could put it all together in a quick network on the next slide. Analysis, and I've stuck to these 8 trials, 8 randomized controlled trials out of all the ones that are available. So it's a simple network that we can look at. We can look at each of the studies, or on aggregate, how did the different treatments compare. Here again, for death being represented in this particular graph, and maybe derive some additional hypothesis that we want to evaluate further. It seemed that our tocilizumab had some activity with respect to minimizing critical disease, but it doesn't seem to do the same thing on the death endpoint. We may ask why, or can we develop some more information to figure out the best intervention as it relates to the disease progression. That's the quick summary I wanted to show. Nevine, I'll hand it back to you.
Thank you so much. And I think interestingly, you might have caught that for Actemra, that is now included in a second phase of recovery. So to your point, Jaap, there will be more data coming shortly. And this shows you that in real time, using Codex, you might be able to actually say, that's really interesting. Does the data exist now? It doesn't. This is so critical. Let's go figure out how to do the trial. So it's a very nice example tying in with some other commentary earlier. Let's go to the next slide. So now you have heard from Jonas and from Jaap all about what's underneath the hood in terms of data and tools. But you know what? That's not enough. To have a thriving community, we need to hear from you as well. And that's why I'm delighted to be getting the questions in the Q&A. I've answered a number of them. I wondered if, as an example of this collaboration, before we go to the panel, Jonas, if I could bring you back in just to describe very quickly, how did we construct the data dictionary? How did we pick which variables? And then we're going straight to the panel.
So the data dictionary was constructed by discussion with the data providers, the pharma companies, reading the protocols, looking at both redacted protocols, redacted SAPs, in order to collect as rich as possible. And given that we're looking at treatments right now, the endpoints were fairly similar. And this was also discussion around kind of what are the covariates we want to include with respect to baseline comorbidities and so on. So I think it was a very rich discussion. I just want to give comments to Peter Mesenbrink, who's put his hat on, and both as a data contributor and as a part of driving the data dictionary within Novartis and throughout the pharma community. So I do think, just to summarize, it's important that we did look at protocols, redacted SAPs, redacted protocols and SAPs to figure out what's in there, put together a proposal, went back to the data computer, tried to kind of enhance it and so on. But I just want to emphasize, it's still a work in progress. It's a continuous improvement is better than delayed perfection. So that's where we're working after.
Wonderful example. And so you've mentioned Peter. So Peter, if we can bring you in, introduce you as a very active member of the community, and very happy to have you with us. Also, Phil Ambery from AstraZeneca, who's been active and helping us with contacts, with ideas, with research proposals. and Ben Kramer, who not only is involved personally with us, but I must say the entire Genentech company, first in with 2 trials of Actemra. Very, very pleased with your engagement personally and as a company. Thank you, all of you. And I put this picture of this nice lady there because I felt like I might be the only lady with all these fine gentlemen, but we can now take the slide down and we'll just go straight into the panel discussion. So the first question that I had for Phil, Peter, and Ben, and we'll start with Peter on this one, is what made you want to get involved with us?
I think the answer to that question is fairly easy, Nevine. I think we were in a situation where the opportunities are unlimited to sort of contribute to the knowledge of COVID-19 as a disease and the potential treatments that may affect it. And it proposed, being a complex disease, we want to be able to explore all data possible and contribute all data that we were able to do so to be able to help the cause. And I think as a statistician, I mean, we're sort of in the best times we've been in our entire lives because we have new research problems and having to tackle issues with data we've never seen before. And I think it's sort of a beautiful opportunity to have a high impact on sort of global health.
So bringing all the skills to bear because there is a global pandemic. Love it. Thank you. And Phil, if I asked you the question, what made you want to add this on top of your workload to activities you undertake?
Thank you, Nevine. And I think pretty much it's a question you never need to ask, really, because actually having been working in the medical profession as a physician for quite a few years now, this is almost the chance of a lifetime to make a difference, actually. And these things only come up probably once in a lifetime. And actually, the chance really to utilize the data to really make a difference to people, not only in America, not only in Europe, but really right across the world, is a chance I was never keen to pass up.
Very good. Thank you, Phil. And Ben, what about you? What made you want to join us?
I think my colleagues have said it well. Very early on, Genentech and Roche, I should say, got involved very quickly in evidence generation, looking for new treatment modalities. And so I think this was, quite honestly, a no-brainer that, as Phil put it, this is a pandemic. This is a serious public health global problem. And this is what we all try to do in the industry, is to try to make a difference and to find new treatments for new diseases where there's unmet needs. And this pandemic was a very specific example of that.
Yeah, so I'm hearing a lot of personal mission stories coupled with organizations that are leaning into this and wanting to make even more of their investment. So I want to ask a question because it might be in the minds of the audience as well. How easy was it to convince your companies to commit to providing data? That's not an easy thing to do. And what do you think that means to all the other organizations who are out there, the recovery team, the Remap CAP team, all those other academic and government funded groups? So let's talk about the arguments for providing data and how did you yourselves engage with your colleagues around that? And I think I'm going to go right back to you, Ben, on this one, and then Peter, and maybe close out with Phil.
Well, I would love to say that I led the charge and that I had a very convincing platform to persuade my colleagues at the company. However, it was they were selling me why we should be doing this. Not that I was against it, but just that I'm just trying to underscore that it was pretty seamless that people thought this was the right thing to do. And we've always talked about how we want to have a platform where we can share data and do it in a collaborative way that will that will help the overall good. And this is what we're doing. And so it was pretty easy. I know that sounds disappointing.
Well, you're lucky, and we're lucky that it was that easy. Thank you. And Peter, what about you at Novartis? How did that go?
I think there's different sort of levels of commitment at at different tiers. At the high levels, I think, and within senior management, it was clear from the beginning that the support was there because they sort of realized sort of our mission from a pandemic perspective. I think we were the first company that actually contributed back when hydroxychloroquine was still sort of being thought of as a therapeutic agent to treat the condition. We were the first to donate our large supply. Obviously, within the teams, we had to sort of change the mindset because the mindset was that the teams sort of have sort of ownership over their data and are sometimes very, very possessive. But I think once we sort of explain to people sort of the past is not the present, and obviously in the global pandemic setting, we have sort of the sort of scientific obligation to sort of share with the scientific community what we know and what we have and what data we have from our studies to help sort of answer some of the scientific questions, which may not be answerable within single trials, but will be answerable from the aggregate data, both at the summary level as well as at the patient level, in data that's being stored in other sources. So once we put it in the proper context, we were very committed. Some took multiple iterations, but then they realized what the impact could be and what we could do with it, and then they were on board.
Good, thank you, thank you. And Phil, what would you say to the academic groups out there who might be sitting on the fence a bit? How would you convince them?
I think I'd say this is a once in a lifetime opportunity to actually share the data. And particularly, as you know, I'm interested in why patients with chronic kidney disease have poor outcomes from COVID, why people with type 2 diabetes have poor outcomes, why people with previous CVD have poor outcomes. And you look across the individual studies, you really can't achieve the power to actually draw any firm conclusions around the phenotype or who is most at risk. And I think it's kind of time to set aside being high up in the ivory tower and actually to actually say, really, for the good of the population, it's time to actually share the data. And if you're sitting on study data from Africa, from Asia, from America, from Europe, it's clearly crucial to help drive improved outcomes in COVID to share the data.
Thank you, Phil. I'm going to use that. So we've had some questions from the audience, and Jonas, there's one here that I want to send to you that has to do with platform trials. We all know that they're very good use of resources at a time like this, but they're rolling studies. So how exactly are we going to go about securing the data?
Yeah, I think that is really important. It kind of also ties into the trial integrity. And we had a real discussion around it in the statistical expert group. And we came to the conclusion, we don't see any really issues with trial integrity for summary level data. Once an arm is graduated and data and results are published, reported in any way, that's fine to share the data. And what type of data could we share? Well, I think we could share beyond what is public as subgroups and so on. That's for summary level data. If you extend it to individual patient level data, that's even more trickier. But we think, well, we came to the conclusion that you can expand it to individual patient level data when the associated control arm has been fully graduated and will not, I'll say it again, not be shared with any other ongoing arm within the platform. And we should only kind of, just to preserve the trial integrity, only include those data elements or whatever is reported in the public domain. So summary level data, it's a bit more than the individual patient level data.
Okay, good. Thank you for that answer. And I must say that we've spoken to nearly all of the study teams or approached them, and that seems to be a place where the community is aligning. There's another question here, which I don't know we've ever discussed. So Jaap, I might be interested in your views, and then we'll see who else from the panel wants to pick it up. There's a question here about how are we going to organize the results, and how are we going to organize all the questions being asked? Because if we have hundreds of researchers worldwide, we're going to need a way to know who's found out what as they looked in the data. So I'll put you on the spot first, Jaap, unless you tell me you want someone else to pick that one up.
Yeah, it's not necessarily something I've thought about quite a bit, so I may push that on that one. But it seems, yeah, it's a very important question with so much information available, so many different subgroups we can look at, so many different time point at which, you know, where the patient is in the progression of the disease and the interaction of the treatment with that. That to organize all the results of people who look at the data would be quite important, and maybe that's the next extension of tools we could build in the environment, where people can push up the results that they have from their evaluations that they've done for other people to interact with and use.
Yeah, I think so. Oh, go ahead, Jonas.
Oh, no, I just want to kind of add to what Jaap said. I think this is a really important question in the sense of building a data research lab in a collaborative way. How do we share the analysis? How do we promote the collaboration between them and so on? If we can organize the results or even the questions before the results come, I think that's going to be a vibrant data research lab. So that's something we need to think about further, but it's a very good question. Thank you.
Excellent question. And Peter, you get the last word on this.
Okay. Yes. I mean, I think it'd be very, in the Workbench sort of environment, it would be very easy, particularly if people are developing their own R-Shiny apps, for example, from a results perspective, so that people then can sort of be able to interact with sort of the results and understand, and perhaps this will also as well generate more hypotheses with the data. So in sort of this in a GitHub sort of like structure, we should be able to incorporate sort of results metadata in there too as well to further enhance the work.
That's a very good point. Okay, so that's how communities work. One question leads to an idea, might lead to a tool or product, and before you know it, we've done so much better than what we set out to do. So thank you for that. Okay, so we have about 3 minutes left, and I think I want to go to around the table quickly. What do you think are the greatest opportunities that ICODA affords for us as we tackle the pandemic? What could we do with this that we couldn't do before? And I'm just going to go with who's on my circle here. Jonas, we're going to start with you.
I think the Workbench is a tremendous opportunity. It's a platform. I like what you said in the beginning. It's a data research lab. It's not just one person or one group coming to one result answer. You're building something within the platform. You get a data repository. You have the tools. You have the collaboration space. And I do think, I want to also emphasize, it's not just for this pandemic. We are setting up ourselves for future pandemics because it's so hard to address something while the pandemic is ongoing. I do think we have the blueprint for the future way of addressing pandemics.
Super, thank you. Ben, you're next.
I think it's been said earlier that when have we had the opportunity to obtain the amount of data that across different sources than in this particular data research platform and the tools. Mm-hmm. Sometimes we have a lot of data, but we don't have the tools to analyze it. And I think the combination of the largesse of the data that we're going to hopefully receive for the data research platform and the tools is really, I think, unprecedented, and I think is required for such a pandemic, and hopefully no more future pandemics, but nonetheless, if it does happen. So I think this is a fantastic opportunity that's unprecedented.
Wonderful, thank you. Phil, what do you think? What can we do now we couldn't do before?
Quite a lot of it's already been said, but I'd make 2 points really. I think one of the key learnings to come out of this is around learning about the power of many, not the power of the few. So
Yeah.
we often end up in an academic world competing across grants against each other and everything. But I think really within the short time that we've had things like the vaccines and the antibodies and other treatments and interventions for COVID-19, we've learned about the power of the many. So the first learning is really, how could we turn the power of the many to solve a lot of the other chronic health problems of not only Europe, America, but really across the world. And the other point, if I'm allowed a second one, is actually we're going to learn quite a lot about the resilience of human beings. And actually we may learn some things which are uncomfortable in terms of the resilience with respect to health services. And I hope that we take those learnings towards whatever comes next in terms of the future, more pandemics or not.
Thank you. So I'm being told that we are at time. So with apologies, first, thank you everybody for your time today in the audience and the panelists and the speakers, but more importantly, thank you all for your work in addressing COVID-19. There is a link here, a reminder of how to get back to the main plenary session where we're going to announce a very exciting challenge cycle coming up. So there's a link in the chat box if you would go ahead and disconnect from here and go back to the plenary. Thank you, everybody.